Bismillahirrahmanirrahim. Assalamu alaikum. Welcome back in the polymer chemistry class again. We have done chapter 6, which is also called lecture 6, and we finish almost the part 3 of that portion of this chapter. Now, I'm repeating this lecture again to just to refresh your memory. And as we know, this part 3 related to the gel permeation chromatography, and we have discussed this lectures very well in the lecture 4 and 5, and the handout is also given for you to understand the concept in more detail. Okay, so this is just a repetition. So as we know that in this introduction we will discuss about the gel permeation chromatography, which will be in more detail, and the mechanism of the gel permeation will be discussed, as well as some data will be analyzed. Okay, so there are different types of liquid chromatography which we use, and you are quite familiar with this HPLC. So here basically we have two different type of liquid chromatography: interactive interactive liquid chromatography, and then you have non-interactive. So when we are talking about the interactive, which means you have some kind of a interaction between the mobile phase and the stationary phase, like adsorption, partitions, ion exchange, etc. Whereas on the non-interactive, we have a method of GPC, which is also called size exclusion chromatography and gel filtration chromatography. So now, as we say, non-interactive, which means that there should not be any kind of interaction, which I have discussed before. So now, if you look into this graph, you can see that we have a range from 10 to the power 1 to 10 to the power 3, like thousands grams per mole. If the molecular mass is like in that range, you can use GC, and then a little bit higher, you use HPLC. And for GPC, or gel permeation chromatography, we use usually the 10,000 and onward, up to million or 10 million, we can use the gel permeation chromatography. So here some nomenclatures we have to discuss before. As the name says, gel permeation chromatography is called GPC. This GPC and size exclusion chromatography, SEC, is all both are same. And the another one is the gel filtration chromatography. Now in gel filtration chromatography, because this is I will discuss this in first, because this you need only some basic informations about that, and then the other one, which is gel permeation chromatography or the size exclusion chromatography, in more detail. So as the name says, gel filtration chromatography, which is very much similar to the GPC, means the mode of working is exactly the same as in GPC, means the non-interactive technique used to characterize the natural polymer in aqueous solution. The natural polymer, like a protein, like DNA, like cellulose, they are used in aqueous solution, and this method can be used. And the common method which are used to pack the column are the hydrophilic gels, and that category we use cross-linked dextranes, cross-linked polyacrylamide. These are the two basically used. Um, Again, the separation in GFC column occurs by the molecular sieving, similar to the GPC. As I told you, because this is the non-interactive technique, so the molecules are actually separated based on their size. That is why this is also called a size exclusion chromatography. But in gel filtration chromatography, we can also use adsorption technique, ion exchange technique, and ion exclusions method. Okay, so now let's talk about what is a GPC system. So in this picture, you can see this is a prototype of a GPC system. So you can see here on the top, you have a pump, and then you have an injector where you inject the samples, and then the column which is packed with the stationary phase, and then you have a detector. So one by one, you can see here the pump. This pump is used to deliver the eluent from the reservoir to a constant volumetric flow rate. The GPC is always an isocratic separation. When we talk about isocratic separation, which means that we are talking about a system in which we have the composition of the solution should not change. If the composition change throughout the process, that is the gradient system, not an isocratic separation technique. Now here in injector, so in the injector is actually have a valve, and it will allow the introduction of the sample here without disturbing the flow rate. And usually we use 200 microliter of the sample which will be injected into this one. And then we have a GPC column, and the GPC column usually packed with crosslink beads of the which used to perform the separation of the molecule based on their sizes. The resolution and the range increase by using the multiple columns. So as you increase the column size, this can increase. And then we have a detector, and the detector, the response usually we get based on the concentration of the polymer molecule which is present in the element from the GPC. So when we are talking about, this is the detector. So in the detector there are different types of detector, and these detectors are named as concentration detector. Among the concentration detector we have differential refractometer, ultraviolet absorption, and evaporating light scattering or mass detector, and infrared detector. So these are different detectors which we use to find out the particular concentration of a particular polymer in the sample. Other is the molecular weight sensitive. So when we are more specifically targeting the how molecular weight, high molecular weight polymer we use these two detector, which is called viscometry, which is sensitive to the molecular weight or the size of the polymer, and the other is the light scattering. So both light scattering and viscometry I have already discussed in the class, and you should know about that. If you forgot, go back to your lecture number 4 or the beginning of the lecture 6 part 1 and part 2, read it again please. Additional system we can have, that's online degasser. So sometime when we use the solvent, they have the air bubble, so that actually interfere in the detector. So we basically require a solvent which is absolutely free of any kind of gas. So the degasser is usually required. So first, sometime we do manually degas the solvent, or sometimes we use an online degasser which do this job. And sometimes we also required several sample to run. So if you do manually, then it requires too much time for you. So auto sampler is also there, so that can take the sample by its own, and all the samples are done automatically. And sometimes our polymer is not soluble to certain solvent at the room temperature, so therefore we require an elevated temperature, and that's why we need oven, and that oven used to increase the temperature of the column. And additionally we also have some specific detector for certain application. And now we are talking about uh, how polymer actually behave in the solution. So we have discussed this also in chapter 4, that in lecture 4, that's the polymer, how the polymer behaves. So the GPC is based on the behavior of polymer molecule in solution. So we know in the solid state a polymer can be considered like spaghettis, uh, which is like a tangled chain. But when you dissolve in the solvent, all the polymer are separated into different entities. But due to the entropic effect, all but most of the rigid of the polymer chain looked like twisted and acquired a shape of the ball that is represented in this picture. So on the left you can see this is like actual polymer, but in the solution it behave like this ball. Okay, so GPC column packing. So as we know that always it's a simple method of separation. So a stationary phase is actually a material which we use. Like when you do a column chromatography, we use silica gel or other other stationary phase. Here also we use, but this is like a cross-linked beads which are used. Mostly we use the polystyrene cross-linked with divinyl benzene, and these beads have a rigid pore structure that remain intact in the presence of solvent. Means it should not react with the solvent in which these beads are going to be used. And the pores inside the beads are responsible for this exclusion of the polymer. So look at this structure. These are the two commercial beads, cross-linked polystyrene with a different pore size. This has a little bit bigger pore size. On the right, you have a smaller pore size, and these are actually responsible for permeating the polymer, different polymer chain. So if we are talking about the permeation of the polymer molecules, I can show you, look it onto the left. This is the polymer chain, red one, which is high molecular weight on the left, and the blue one is little medium size, and the green one is really small. So if this is the polymer bead which we have inside the chain, now if we see more closer to this, you can see the small holes there this is called the pores. So what happened when you put the polymer in the column, when it passed through the column, because of the larger size, it cannot go inside the pores, so it's actually permeated, it is excluded, so it can bypass these pores and directly go out. On the other hand, some chain which are medium in size, they can partially permeate it, so partially it get in and come out, but the smaller one completely penetrate into, and that's delay its exclusion from the column. So this is the basic principle for uh, for the separations of the polymers. So the polymers are separated based on their different sizes. So as the size increases, it cannot go inside the pore, and the polymer will come out immediately from the column or excluded before the small molecules. Okay, so this is the basic principle of this. So now in this picture, the phenomena can be shown. So you look at here on the top right corner, I have a polymer sample which have three different types of molecular weight, and this is in the middle. The column is packed with the pores, so it passed through this one. Since the high molecular weight cannot go inside the chain, so it can pass through very quickly and come out earlier. So in this picture on the left, you can see that the detector shows a peak on the left, which is high molecular weight, so it comes first, which means come out a little bit earlier with the low retention time, or less amount of volume will be used. On the right is the polymer which come out at the last, will be the smaller molecule, because it gets completely permeated into the small holes. So this is basically the mechanism. So you can read this one, the information is mentioned here later on. And so when you do, so the summary is here. The molecular size elution takes place with the molecular size, and the larger molecule will be eluted first, and then the small molecule the last one. So in this picture again, you can see here, so the top one is showing the different peaks for a different polymer with a different molecular weight. So the first one on the left, where the cursor is right now, it's a high molecular weight, and then gradually the molecular weight decreases, and then you get the solvent peak. So solvent peak is usually comes after the 30 ml. So after the 30 ml, usually you get the solvent peak. So that's why when you run the GPC, or also depends upon what solvent you are using. So when you are using the THF, tetrahydrofuran, this is the solvent peak usually comes at this point. So that's a peak. What we are showing here is the different range of the polymer which can be shown. So as a result of the GP separation mechanism, polymer molecule eluted from the column in order of size in solution. Larger molecule elute first, the smaller eluted the last. So this red one is showing the larger molecule that come first, and as the molecular weight decreases, and the last you get the smaller one. So here we have different nomenclatures used: V naught, VT, VP, and what are these? This will be explained in the next few slides. So the separation is purely a physical partition. There is no interaction or binding, and the separation is isocratic. As I told you, isocratic means throughout the separation process, the concentration of the solutions, the composition of the solution remains same, it does not change. Okay. So so as I'm talking about whenever the GPC is used, so the more in the GPC we always required the column to be packed with some materials which is called the packing material, and packing materials are usually a porous particle, a cross-linked bead, and that pores should not interact with the solvent which we are using to dissolve the polymer. Okay, so and this is usually prepared by celery packing technique, and at the pressure of 2000 psi. The column which we use have the inner diameter of 7 to 8 millimeter and 250 to 600 millimeter in length. As mentioned in the previous slide, we can also increase the length of the column for the better resolution. So which means that we can add on an extra column or two along with the normal GPC machine. Okay, and here the terminology means, so V naught is the exclusion volume upper limit, means after that no matter how larger the molecular weight you have, all molecule will be eluted at the same place. So that is called the upper limit. I will take you back to the previous slide. Look at here. Here is, you can see all the, this line is going straight on the top, which means that as the molecular weight is, there is a certain range of the molecular weight. After that there is no more change takes place. Similarly, in the lower side, the molecular weight are so small that all will go in, penetrate, all will be permeated in the pores, and therefore this will not be separated. They all eluted at the same place. So this is called your exclusion volume upper limit, which is the upper limit, and then you have total permeation volume VT, which is the lower molecular weight limit. So there is a particular range which is also called the VP, working resolution resolving range of the molecular weight. So this is the area where we can trust the molecular weight of the polymer weight which we want to separate, neither the top limit or the lower limit. So if your polymer which you want to separate falling into upper limit or lower limit, that means the separation is not taking place well, and we have to add another column or to increase the calibration limit. Okay, so for the calibrations, so all these terminologies, you know, MN, MW, MV, I have discussed this, and polydispersity index or PDI. So GPC is a machine that can give you all these things: MN, MW, MV, MP, and the polydispersity at the same place. Before that, I have discussed the method by which you can get only MN, MW, or MV. But now in this GPC machine, we can get all these information at one place. But as I mentioned to you, you required a calibration. So there must be a calibration will be required to make a range of the polymer molecular weight in which you have a certain upper limit and lower limit, and your polymer should fall into that category. So most commonly used polymer calibrants are polystyrene. So polystyrene, we have a standard neuromolecular weight distributed polystyrene sample which can be used for calibrating the GPC machine. And the solvent we can use THF, toluene, chloroform, and TCB. Similarly, polymethyl methacrylate can also be used, but the solvents are different. The name are mentioned to you here. And similarly, for polyethylene oxide or polyethylene glycol can also be used in the aqueous. We can use aqueous eluent and DMF or DMS. So remember, these polystyrene, methyl methacrylate, and polyethylene glycol should be a cross-linked, and they are used as cross-linked beads, and they are used in the column. So what I was talking about, the error in the due to the limited calibration region. So if you look at here, this is the only place where we can trust the straight line here. We can trust the molecular weight of the polymer, and here you have the upper limit, which means that as the molecular weight increases, all polymer will exclude at the same place. Same for the lower range. So if your molecular weight, if the molecular weight of the polymer less than 2k in this particular example, or more than 20k, then this will not be a good calibration curve. In that case, we have to expand our range for calibration. Okay, that is why we need a universal calibration. And how this universal calibrations, why do we need it? And because a major problem with calibrating a GPC column for a particular polymer. So as we have discussed in the previous slide, that we use a calibrator like a polystyrene, or we use polymethyl methacrylate, or polyethylene glycol, but we need a very narrow molecular weight distributed polymer. But unfortunately, only a few standards are available in the market. Mostly we have available is polystyrene from 600 to 2.5 million um, molecular weight range. And but the problem is that, however, if you are dealing with some other polymer, then the polystyrene molecular weight which is obtained will have be in comparison to polystyrene standard, will be in serious error. So ideally, whatever the type of polymer you're going to separate, or you want to find the molecular weight of that particular polymer, you should calibrate the column by using the standard of that particular nature of the polymer. But as I told you, usually the standard material are very rarely available, because we need a low polydispersity index polymer, very close to 1, and only polystyrene, in some cases polymethyl methacrylates are available. So usually we get the molecular weight of the other polymer in comparison to polystyrene standard. That's why sometimes the material, the result which is obtained, cannot be completely trusted, may have some serious error. That is why we need a universal calibration that can accommodate different types of polymer which are entirely different from each other. Okay, that's a reason for fine requirement of the universal calibration. So what are the basic parameters for the universal calibration? So universal calibration is based on the observations that the product of intrinsic viscosity, we have discussed this in last lecture, and the molecular weight is independent of the polymer type. So if you have intrinsic viscosity and molecular weight, and its product is independent of the type of polymer, so this is the basic thing which we can use, and this product is called the universal calibration parameter. So if it is asked what is the universal calibration parameter, so the product of the intrinsic viscosity and molecular weight is the universal calibration parameters. Now let's see how this is going to work. So if you plot log of the universal calibrations parameters against the elution volume means the volume which is required to show one peak on the GPC machine, and the solvent is used tetrahydrofurane in this case. So then when we plot this one, this will give you a single curve approximately linear, and it is shown in the figure 2.11. And this is the picture you can see here. This is pretty much a straight line, but you see an upward turn in the higher molecular weight range. The reason is exactly the same as I told you before. There is an upper limit, means all the molecular weight size after a certain point, they just cannot they are completely excluded. So this is the upper limit. So this is the universal calibration. So any polymer from here to this range can be obtained. So look at here, all these polymers names are mentioned here, and these can be molecular weight can be find out with the help of this calibration graph. So, so this approximately linear plot obtained for a widely disparate means different or unequal or unrelated polymer group of polymers. So, the log of the intrinsic viscosity and molecular weight may be considered a constant for all polymer, provided that the following should be same. we can only use this one, universal calibration parameter, under these conditions. Same column, solvent should be same, temperature should be same, and the elution volume at the same amount of volume should be required to exclude that particular polymer. Then it is applicable, otherwise it's not. If any of these four are not same, these calibration graph cannot be used. Okay, so based on that, suppose if we have our reference polymer, we are using polystyrene. So the polymer 1 is the is the our calibrating polymer. In this case is polystyrene, and polymer 2 is the other polymer which we want to fractionate and find out the molecular weight. So we can write this one. The product for first polymer will be intrinsic viscosity time molecular weight of the first polymer, and similarly for the second case. And from the this equations, MHS equation, we can find the formula like that, which we have discussed before. And when you are combining these two equations by taking the log, of course we will end up with this equation, which I named here as equation 1. This is equation 1, and this is the equation 1. You got it. And now here, if you look at here, we have K1 and K2 and A1 and A2. These are the constant which we can get from the polymer handbook for different temperature and for different solvent. And therefore, and as I told you, M1 is the molecular mass of the standard which we use. So we know which standard which we are using. We can find the value of K1, K2, A1, A2 from the polymer handbook and the table given in the book. So if the questions given to you for finding the molecular weight of unknown polymer using this equation, all these informations you have to get it from the table which is given in the book. Okay, or of course this will be provided in the exam or in the assignment. If this question given in the assignment, you have to find the value from the book. So so in that case, in order to find the molecular weight at a given retention volume, the column must first be calibrated. So as I told you how to calibrate before, of course the solvent and temperature and the column should be same. So we will get the similar graph as we shown before. It's exactly the same as in the figure 2.11. See here you have an upward turn here. This is because of the upper limit, and this is the high molecular weight limit. Means after certain point from where this upturn start, after that molecular weight, for example, if this is like 100,000, so after 100,000 no more molecular weight will be reliable, because all will be eluted at the same place. And here this is the uh, lower molecular weight limit. So let's say the loss is 2000, then less than 2000 will not be acceptable result, because all will be eluted at the same place. So this graph is very very important, especially this upturn and the lower turn. And you must know why you have this upturn and lower turn, and why the results are not reliable on that place, and why we have this upturn and lower turn. And you should be able to explain this thing. And this is probably the answer for the last question in the assignment. You should concentrate on this slide and explanation which I mentioned to you, and this will help you in writing the answer for the last question. Okay, so as we discussed this one, uh, so again it's a linear graph. So if you see here, the linear graph means this is the linear space. So the any molecular weight in this range are reliable, not the upturn and the lower turn, as I mentioned to you. That's explain again here. And this is called the deviation from the linearity at the higher molecular weight and the lower molecular, and both you should be able to explain why. Okay, so the constant, as I told you, K and A are normally obtained from a polymer handbook, and then you can substitute all these value. I already explained M1, K and A, and then you can find the M2 value. Okay, now nowadays in the DPC machine we are using light scattering detector. So once you start using the light scattering detector or better light scattering detector, then because the light scattering detector purely depend upon the size of the polymer polymer chain. That's why in future on the more and more use of the light scattering detector will actually eliminate the need of the universal calibration. So therefore you don't need to use universal calibration if you have a light scattering detector in GPC machine. So without that you have to rely on the universal calibration. Okay, now how to interpret the chromatogram. So look at here in this graph. This is the graph it's look like. So you see some baseline here, which is called the dead space, and then you see a peak at a certain minute or certain volume. If you are using the minute on the on the x-axis, then it's called retention time, and if you are using the volume, then it's called retention volume. Okay, both can be used. So this is your samples peak, and then after certain point you will see a downward peak which is called a system peak. After that you have a big peak which is called the flow rate. So the straight peak will show you have a stable flow rate, and this is the internal marker. If this peak is not straight, which means your flow rate was not stable, then your result will not be reliable. After that all the peaks are belongs to the small molecule, your solvent, or any small molecule impurities in the system. Okay, and these information usually mention on the GPC chromatogram, like what column you are using. It is very important to mention what is the size of the beads you use, what element means the solvent you use, what is the flow rate were used, what was the flow rate, sorry, I missed something. Yeah, flow rate and what kind of detector. So here we use differential refractive index DRI detector we have used here. And if you use RI detector or light scattering detector, that name should be mentioned here. And that's how you read your graph. So when you elude the peak, sometimes so the important thing is that the peak should be, for example, in this case the peak is going up, and this red line showing that the peak should come down and touch the baseline. If that's not the case, then this result is not reliable. Why it is happening? Because two of the polymer are pretty much identical or very close in molecular weight to each other. That's why they are co-eluted, and which means they are not completely touching the baseline. Look at here again. So this, if it is the normal two peaks, so this peak goes down here on the red, and then the second peak will go like that, and both are separated. But now here, so this result is not completely separated. I mean, like in this case the molecular weight are not completely separated because they have very much close to each other. So under that condition, if you still want to do the calculation, yes, you can do the calculation as shown here. This is peak 1. So from here you can get the molecular weight of peak 1, and from here you can get the molecular weight of peak 2. But this, the polydispersity, which means MW over Mn, is not reliable because both peak are not completely resolved into the baseline. So ideally, we should have a system in which the peak are completely resolved and touch the baseline. If not, then the result is not reliable. So what type of question could be asked? We can give you a different speak in the exam, and then you have to just just you have to tell if this can be resolved or it can be used to figure it out the molecular weight or not. As I told you, yes, you can figure it out the molecular weight, but the molecular weight distribution or the polydispersity index cannot, because it did not completely resolve and touch to the baseline. Okay, now sometime in this, the last slide is the, if you see here, there are two different comparisons given to you. On the left you see the peaks are much broader as compared to the peak on the right. Why so? This is based on the two things, like depends upon the pore size of the material which are used in the in the column. So if you have a smaller pore size, if you have a smaller pore size, then the peak will be very thin and nicely and sharp and nicely eluted. On the other hand, if the pore size is big, then you have a broader peak. So in order, which means that it is always better to use a column in which you have a lower pore size, and that will give you a better separation result compared to the column in which you have high I mean like the pore size or higher size of the pore you use. Okay, and that's it about the gel permeation chromatography, and I hope you understand this lecture now. So please go through the slides which I have already given to you, and watch this video, and then try to solve the questions related to the GPC. In the next video I will talk about the next topic, which is about the types of polymer. Hope you understand this one, and please submit your assignment on time. Thank you.